My screen now?
Looks fine.
Good. Okay, so my name is Iren Löhr. I'm a clinical microbiologist at Stavanger University Hospital in Norway and currently the chair of Nordicast. So in this talk I will focus on ESBLCARBA, mechanisms of resistance and detection. So what is ESBLCARBA? I think you're all familiar with this. In the meantime, ESBLCARBA are acquired carbapennamases, which are enzymes that confer resistance to all or most beta-lactams. As always, there are exceptions, and for instance, OXA48 is a bit tricky. We'll come back to that one. There are three classes of ESBLCARBA. Class A, Klebsiella pneumonia carbapennamases, abbreviated to KPC. Class B, the metallobeta-lactamases, or the MBLs, divided into different families, and the most important families are the NDMs, the VINs, and the IMPs. And the Class C carbapennamases are the OXA-carbapennamases, where the OXA48-like enzymes are of importance among Enterobacteriales, for instance, and OXA23-like among Acinetobacter. So which bacteria harbor carbapennamases? They are found in Enterobacteriales, like Klebsiella pneumonia, E. coli, Enterobacter, and so on. Therefore, we also call them carbapennamase producing Enterobacteriales, with the abbreviation CPE, which I think most of you are familiar with as well. Also, we find ESBLCARBA enzymes in Pseudomonas and Acinetobacter species. So, ESBLCARBA and spectrum of activity. It starts to get more complicated already. Ørjan Samuelsen from the reference lab CORES in Tromsø borrowed me this slide, which is a nice overview, I think, over the different ESBLCARBA classes and different enzymes within the classes and their spectrum of activity. And as you can see, there is some variation, but in general, we could say that all ESBLCARBA enzymes confer resistance to penicillins, first and second generation cephalosporins, and at least mostly the carbapennams. Whereas there is, as you can see, varying activity against the extended spectrum cephalosporins, like cefotaxime, for instance, and also against the monobactam espernan. ESBLCARBA, I got some slides or data also from Ørjan in Tromsø. These are unpublished data, which I think will be a part of the NORM report this year, data from 2007 to 2020, where you can see on the picture to the left the different species and number of isolates in total with confirmed ESBLCARBA detected at the reference lab in Tromsø. And you can see there has been an increasing number of isolates every year, except 2020, which I think was a very special year due to the pandemic. And what this picture also shows is that the most dominating species harboring ESBLCARBA detected in Norway are E. coli on the first place and Klebsiella pneumonia on the second place. The figure to the right shows which enzymes they harbor. And as you can see, NDMs and OXA48-like enzymes are most commonly detected. And just a note is that all isolates having reduced susceptibility or resistance, at least phenotypical resistance to carbapenems, aren't necessarily ESBLCARBA producers. So in Norway, we send all isolates where ESBLCARBA is suspected to the reference lab for further confirmation. And they found at least in 2020 that approximately 1/4 of the isolates, of the Enterobacterialis isolates, with reduced susceptibility to meropenem were ESBLCARBA producers, whereas the rest had other mechanisms. And which other mechanisms? In Enterobacterialis, of course, they can produce carbapenemases, but they can also produce regular ESBLs, ESBLA, plasmid-mediated AMP-C, or chromosomal AMP-C, and this combined with impaired permeability causes carbapenem resistance in very many of the isolates. In E. coli and Klebsiella, the combination of ESBLA and impaired permeability is quite common, and in Enterobacter, for instance, it's the combination of chromosomal AMP-C and impaired permeability, which is the most common. So this is also just a figure borrowed from Iren, showing the different mechanisms, reduced permeability, efflux, and also mutations in the pentylene binding protein can cause resistance to carbapenems. But these all mechanisms together with different ESBL enzymes often cause carbapenem resistance. So when it comes to ESBLCARBA, these are also data from Norway in Pseudomonas and Acinetobacter. When we suspect that we have isolates which are carbapenemase producing, we send them to the reference lab in Tromsø as well. And as you can see from the figure to the left, ESBLCARBA is confirmed in lower numbers of Pseudomonas and Acinetobacter compared to Enterobacterialis, but there is an increasing trend from year to year also for these two species. I couldn't find a figure showing which enzymes these are, but the figure shows that ESBLCARBA is more common among Acinetobacter compared to Pseudomonas in Norway. And when we look into which enzymes we find within these species, the 5 isolates detected in 2019 were NDM, VIM, and IMP. Whereas in Acinetobacter baumannii, most isolates harbored OXA23, a few OXA72, NDM, and even a novel NDM. And in Acinetobacter species, 6 isolates were detected with ESBLCARBA. 4 of them had NDM1, and 2 of them 2 other OXA variants. When you look at the figure to the right, I think this nicely illustrates, and these is also from the norm. Both figures are from the NormVet report from 2019. And the figure to the right shows the prevalence of resistance in Pseudomonas aeruginosa in blood culture isolates from year to year. And as you can see, the levels of resistance to both meropenem and imipenem. Imipenem is the red line here, I think, and meropenem, the blue, are quite high compared to the numbers of isolates with confirmed carbapenemases. And the reason for this is that impaired permeability or efflux combined with the other enzymes in some cases is the most common cause of carbapenem resistance in Pseudomonas. So earlier this year, Nordicast finally published a new methods document on detection of carbapenemases. You may have seen this already. I think it was published back in February. So I will go briefly through that. This is a table included in the document, similar to the table I showed earlier, but it shows gives an overview of the most common acquired carbapennamases and the different enzymes and classes, and also which inhibitors are active within the different classes. Yes, detection of ESBLCARBA is not so easy. There are many challenges. We have just spoken from the different mechanisms. Every resistant isolate doesn't harbor ESBLCARBA. And then there is the challenge with the diversity of enzymes and the fact that some ESBLCARBA remain clinically susceptible to carbapennams. And this cartoon shows that, I think, also quite nicely, that especially for ESBLCARBA, CARBA class D, you may end up having an isolate which only compares clinical resistance to penicillins, and in some cases only to carbapennams, and that makes them especially challenging to detect. For CARBA class A and B, it's easier. They are often resistant to third-generation cephalosporins, and the CARBA A enzymes are always I or R to aspirinam as well. But this is the reason, and especially the problem with the CARBA D class, the OXA enzymes, makes it important to, or this makes it obvious that we need something else than the clinical breakpoints to find all carbapennamase producing isolates. So that is the reason why we need a specific screening breakpoint for the Enterobacteriales. This figure shows that if you would rely on the clinical breakpoints for Enterobacteriales, which are 16 and 22 millimeters, there would be a lot of isolates here, over 22 millimeter with disc diffusion, which would not be detected. And if you look at the colors, the purple parts of them, the purple isolates are isolates which are OXA48 producers, for instance. So you would miss a lot of the OXA48 producers. And you would also, if you see there are some blue and red dots here as well, meaning that you would miss some of the NDM and VIN producers as well, for instance. While if you use the screening breakpoint of 28 millimeter and say you capture most of the resistant population. So therefore, we do have a meropenem screening cutoff for detection of ESBLCARBA in Enterobacteriales. 28 millimeter if you use disc diffusion, and MIC above 125 if you use E-test or microbroth dilution. All isolates with values below or above these cutoffs should be tested for the presence of ESBLCARBA using phenotypic and genotypic methods. And as also stated in our methods document, additional testing should not delay reporting of AST results according to the clinical breakpoints to the clinicians. Which results should be reported as tested, but you could and should add a comment that ESBLCARBA is suspected and that might have implication for infection control, for instance. And detection of one could ask, why is it then so important to detect the resistance mechanisms? And I think we all know, working in hospital labs, that it's important for infection control and of course also for surveillance and public health purposes. But the take-home message here is that it should not delay reporting of the AST results. This is the new algorithm for detection of ESBLCARBA in Enterobacterialis, published in the Nordicast table version 11 and also included in the new methods document. So what is new in this compared to earlier versions? It looks somehow similar, but we have added some boxes, and that's that you can, if you have an isolate with meropenem, with a meropenem zone less than 28 millimeters, and this piptazo is above 20, you should go on with further testing. And in addition to using phenotypical testing, as we have recommended before in our flowchart, we have added a box which says you could also go ahead and do direct detection using NAT, meaning PCR or something, another nucleic acid test, or even a lateral flow test. You could also go ahead and with direct detection using biochemical test, or if you, but then we emphasize with this arrow that you should verify the result, a positive result from a biochemical test with an additional test, for instance, a PCR, lateral flow test, or whole genome sequencing. Whereas we say for the PCR or lateral flow test, this is reliable enough. It could be the final conclusion. Anyhow, in some countries, for instance, in Norway, we are to send all isolates where we suspect or even have confirmed locally an ESBLCARBA, we are to send them to the reference lab for final verification before they are notified. What is also new in the flowchart this year regarding phenotypical detection, there are no changes except that we have put in an additional box regarding temocillin, emphasizing that we can't really rely on temocillin to detect all OXA variants. So OXA48-like enzymes will normally be resistant to temocillin, but there are other variants, for instance, OXA244, which are not resistant to temocillin. Therefore, you can, in a way, use temocillin to say, well, if it's resistant, it's probably an OXA48 or something similar. But if it's susceptible, you will need to go on with additional testing. So, phenotypic detection. I think most labs still doing phenotypic detection of ESBLCARBA would use the combination discs. This is a picture with the Roscoe discs, which we also use in our lab in Stavanger, and I think they work very well. It's a combination of one disc with only meropenem, and then you have one disc with meropenem boronic acid, one with meropenem and cloxacillin, one with meropenem and DPA, and finally the temocillin disc. And if you, for instance, like on this picture, if you read a difference, you read the zones, and if you have, in this case, a difference of more than 5 millimeter between the meropenem alone disc and the meropenem DP test, it would indicate that this is an MBL producing isolate. So I think these discs are still very useful if a phenotypic testing is what you prefer to use in your lab. I borrowed these slides from Örjan. Just a few examples. This is an ESBLCARBA A, I guess KPC producing Klebsiella pneumonia. And as you can see on the plate down to the right, this nicely shows a positive test where the with a zone difference between the meropenem disc and the meropenem disc with boronic acid. And PCR also confirmed this with a positive KPC result. This is a, oh, sorry. This is, I can't see the top of my screen. So this is an E. coli, I think, with a suspected MBL. The plate down to the right again shows a nice zone difference between meropenem and meropenem DP, and PCR also confirmed MDM in this isolate. And finally, one example showing a Klebsiella pneumonia with OXA48, where temocillin was resistant. I didn't comment on this, but for all these examples, you can see, at least for this OXA48 producing isolate, the gradient test doesn't give any additional information. For NDM, it did, but for the KPC, both tests were inconclusive, leading to my conclusion that the discs are maybe more reliable and more useful for most cases. So briefly, ESBL-CARBA can also be detected directly, as we said, for instance, with a PCR. And if you do this, you should always consider which enzymes or genes to include in your PCR, and for Enterobacterialis, that should be OXA48, NDM, VIM, and KPC. And also consider if the test is negative, but the isolate still, for some reason, suspected to be carbapenemase producing, consider further testing or with another assay or a lateral flow test, or even whole genome sequencing if that's available to you. Then we have the lateral flow assays. I just picked one example here. This is the test we are using in our lab, the CORIS resist test, which detects OXA48, KPC, and VIM and IM. And I think this test works really nice in most cases and gives a reliable result. And there are many other tests on the market as well. Finally, there are lots of different biochemical tests and assays available, which can also be used, but most of them are less reliable than a PCR or a lateral flow test, and that's the reason why we also say if you use such assays, you should always confirm your result. So to summarize our recommendation, which is also stated in the methods document for ESBL detection in Enterobacterialis, we support UCAST's recommendation of using meropenem instead of ertapenem, for instance. Meropenem gives the best balance of sensitivity and specificity when screening for reduced susceptibility to carbapenems in Enterobacterialis. For labs using disc diffusion, there is no added benefit in determining the meropenem MIC. And remember, isolates with suspected carbapenemase production should be reported as tested according to clinical breakpoints, with a warning or a comment about possible CPE whilst confirmation testing is carried out. If you perform direct testing, be careful that your PCR or lateral flow test include the most important carbapenemases. And again, if the test is negative, but there is a good reason to suspect carbapenemase production, consider further testing or whole genome sequencing. Just one slide on detection of ESBLCARBA in Pseudomonas. This is part of this is the flowchart from the Nordicast breakpoint table version 11, where we also did just a small modification this year, including a box for direct detection using NAT or lateral flow tests or even biochemical tests. In Pseudomonas, the MBLE test, I think, is probably a lot used, but it does have, yeah, it's not always as good as it should be, and probably PCR or a lateral flow test would be more helpful. And again, if you use a PCR or lateral flow assay, remember to include the most important genes, and for Pseudomonas that would be Vim, IMP, and NDM. Acinetobacter, if you have an isolate which would reduce the susceptibility to meropenem, you should suspect ESBLCARBA, of course. And for Acinetobacter, the MBLE test doesn't really work good, so we recommend, in line with the UCAST, that a PCR should be used. And again, remember to include the most important genes, and for Acinetobacter that would include OXA23 and some other OXA variants. Yeah. If you need more information, see our website and our method document, which is at least updated for Enterobacteriales, and we aim to update our Pseudomonas document as well. And then there's a lot of information, of course, if you have interest in reading the UCAST guidelines for detection of resistance mechanisms, which I can highly recommend you to do. And finally, thank you for your attention. Thanks to the Nordicast group, especially the gram-negative group, and also to Örjan in Tromsø for always contributing with good with information, and in this case also with some slides and pictures. So thank you all. Ulrik, I think I will unshare.
And I can tell you that there is a question in the chat for you, Iren, yes, on the ESBLCARBA. If you have an ESBLCARBA isolate that is initially susceptible to carbapenems, do you have any information on the risk for this one to turn into a resistant isolate during a treatment, or that it can be resistance during treatment?
Yeah, I guess the question is similar to if the same thing happens in meropenem resistance isolate, for instance, like we are used to in Enterobacter, where we know if you have a chromosomal MC that can result in more resistance during treatment. it looks like Christian have some input on that, but I guess it depends on the mechanism. If you have the ESBLCARBA enzymes, they are expressed if they are there normally. But if you have other mechanisms, it might be different.
Yeah, do you want me to comment a bit more on that? It's, I think, difficult to set a certain percentage on the risk, as we can do for Enterobacter, for instance, where you can say that the risk is around 20%. I think it probably will differ between different classes of carbapenemases, and also with the resistance level at the outset of therapy. Perhaps there will not be so much more data on this also, because I think if it has been confirmed that you have a strain of such kinds, I think people will nowadays choose other therapies, which will be more covered in the next presentation. But personally, I think if you have a low MIC, it should work to have a fairly low risk of selecting resistance, at least with OXA48. If you treat with meropenem in a good dose. Many would advocate combination therapy, but I think mostly people will switch to some of the novel antimicrobials that are covered in the next talk.
Thank you.
There was also another question for you.
Yeah, I see that.
Yeah.
I think it's Jørgen. I may have been unclear, but I meant to say that if you do a NAT or a PCR, for instance, or a lateral flow test, there is normally no need to verify that result. So that's what the flow chart shows. If you do a biochemical test, you should confirm that with, for instance, a lateral flow test or a PCR.
I guess the point was maybe also that the NAAT and the lateral flow mostly cover the same variants, right? So in case you believe it to be something more uncommon, it usually is, unless you have a specially adapted NAAT panel, it would be more like whole genome sequencing, which I think was on your slides.
Yeah. Okay, I see people have already answered to my first question, and that's very good, because now we are maybe running a bit late. What are the most common mechanisms for resistance to meropenem in Enterobacteriales in the Nordic context? And I see most people have said that could be acquired carbapenemases or ESBLs combined with reduced permeability, or chromosomal MC combined with reduced permeability. And I guess that's a good guess for most of you. In fact, I mentioned some results from the reference lab in Tromsø, where they find that in Enterobacteriales, only roughly a fourth of the isolates which are meropenem resistant do have carbapenemases. So ESBLs combined with reduced permeability are even more common, at least in some species like E. coli and Klebsiella. And of course, in Enterobacter, for instance, chromosomal abscess with reduced permeability is probably the most common reason for meropenem resistance. Okay, let's take the next. Probably an easy one. Yes, I think I am happy about this. The clinical breakpoints used for ESBLCARBA screening in Enterobacteriales, and most of you say no, which is correct. We do have a specific screening breakpoint, which should be used. So I think we can go to the next question. Which methods do you use in your lab to detect ESBLCARBA in Enterobacteriales? Yeah. So people do different things, and it looks like most people still use phenotypic detection with disk diffusion, but there are also a lot of labs using direct detection by PCR or lateral flow tests, it seems like. And some are using whole genome sequence, I guess probably not for the primary screening, but maybe for confirmation, I guess. So this looks nice. And this is all according to what we also would recommend, I guess. I think we can go to the last question. How would you report an E. coli isolate susceptible to meropenem according to the clinical breakpoint, but with OXA48 detected by PCR? And most people say they would report S with a comment, which I think is a very good answer. There are some saying they would report resistant. That's maybe to ensure that, yeah, because you're unsure about the effect of the gene detected. But according to the guidelines, I would say S with a comment would be the most correct answer here. Okay, I think that's it.